There are multiple ways to identify microorganisms. One of the most common techniques is known as identification by phenotype. And in identifying by phenotype, we recognize microorganisms or identify them based on their shape or morphology, and also their metabolism, or the results of biochemical tests using what we call selective media and differential media. More recent advances both in immunology and in genetics have led to techniques that allow us to identify microorganisms based on their response to an antibody or the antibody's response to the microorganism, and these are known as serological techniques. And then genotyping based on the sequence of what's known as the 16S rDNA. And this is a piece of DNA that encodes the 16S rRNA, which is a subunit of the ribosome. In the 1970s, the use of 16S rDNA began and allowed us to regroup the tree of life into bacteria, archaea, and the eukaryotes. We also can sequence individual genes, or we can take an organism's DNA, cut it up with enzymes, and then separate those fragments of DNA on a gel through a process known as DNA gel electrophoresis. I want to take a minute to distinguish the words species and strain, because not only do we use these oftentimes in the laboratory, but we also use these terms often in class. A species is a population of cells with similar characteristics, and a strain are genetically different cells within a same species. In other words, E. coli, or Escherichia coli, is a type of bacterium, a species of bacterium. But there are individual strains that have slightly different characteristics from one another. For example, K12 and strain B are non-pathogenic strains of E. coli that have different genetic makeups. O157H7, on the other hand, has another different genetic makeup, which includes some very specific genes that are required for its pathogenicity. Now, oftentimes we're using multiple different types of tests to identify a microorganism. No single one test is going to be enough to help us identify a species, let alone a strain of bacteria. And the way that we can consider looking at microorganisms or identifying them is by using what's known as a dichotomous key. A dichotomous key is essentially a series of yes or no questions that can be used to distinguish microorganisms from one another. Now, many of you may recognize this game board right here. This is the classic game Guess Who, in which you have a number of different faces, 24 in total, and each of these individuals on here has a special group of characteristics that helps to differentiate them from all the other members on the board. So let's take a look at an example of a dichotomous key using this game Guess Who. So let's say that we ask the question, do you wear glasses? If the answer is yes, that leaves us with 4 individuals: Sam, Claire, Tom, and Joe. If the next question was, are you bald? Again, this is a yes or no question. And if the answer was no, that would rule out Sam and Tom as being the person, but rule in or make it possible that Claire and Joe are still potential candidates for who the other person in the game has. And then the last question, do you wear a hat? And if the answer is yes, it's Claire. If the answer is no, the individual would be Joe. So Guess Who is a great analogy for a dichotomous key. Now here's another example where we can use a dichotomous key to distinguish microorganisms from each other based on their growth characteristics. In this example here, we have 3 different types of gram-positive cocci bacteria: Staphylococcus epidermidis, Staphylococcus aureus, and Micrococcus luteus. And you'll see in this table, I've put yes or no answers to whether they are able or capable of growing in 6% salt solution, or whether they have the ability to ferment the carbon source mannitol. What I'm going to allow you to do is take a minute to go ahead and try to draw a dichotomous key distinguishing these three microorganisms from each other based on these two characteristics. Pause the video now. So now that you've had a chance to perform that dichotomous key, let's take a look at an example of a correct answer here. So the first question we might ask is whether the organism can grow in 6% salt solution, or NaCl. If the answer is no, then the microorganism is Micrococcus luteus. If the answer is yes, that leaves us with two potential microorganisms, Staph epi and Staph aureus. So then our next question from the yes might be, can the microorganism ferment mannitol? If the answer is no, then the organism is Staph epidermidis. If the answer is yes, then the microorganism is Staph aureus. So simply by asking these two questions alone, we're able to distinguish these three microorganisms from each other. Let me now take a minute or two to introduce you to the last two techniques of distinguishing organisms or identifying them and classifying them. One of these techniques is using genetic sequencing or genome sequencing. These days we're at a point in biology where whole genomes can be sequenced for a very, very cheap price. One of the regions that we typically look at is the 16S ribosomal DNA sequence. In eukaryotes, this is the 18S sequence. Now this sequence is highly conserved, which makes it almost act like a molecular clock. But there are a couple of variable regions, and you can see those listed here. They are colored and listed with the V representing variable 1, 2, so on and so forth. And by sequencing or determining the sequence of these individual variable regions, we can identify at least the genus, if not the species, of a microorganism. By comparing the sequences between different bacteria with each other, what we can do is come up with a rough sketch or a rough map of how related the microorganisms are to each other. So for example, between species 1 and species 2 listed here, you notice that there are two differences, a GC and a CT, but the A, the C, the T, and the A are all the same, and so there are two differences. And if we look at this between the different microorganisms, we find that between 1 and 3 there are 4 differences, between 3 and 4 there are only 2 differences. And what we can say then with pretty good confidence is that 3 and 4, because there are only 2 nucleotides that distinguish them from each other, are more similar to each other than they are to 1 and 2, which also differ from each other by 2 nucleotides. By doing this sort of analysis then, we can come up with maps or trees to help model the evolutionary relationship between microorganisms. The last technique we'll talk about is serology, and the term serology comes from serum. Specifically, what we're talking about are antibodies that recognize a specific pathogen. The antibodies, we know which pathogen they're going to identify, and so this is essentially used as a yes or no answer that a pathogen is a specific strain of microorganism. Let me give you an example here. In the slide agglutination assay, we actually have physically a glass slide with 2 areas to put our bacterium. And in this particular test, what we do is we add antibody to the bacterium and look for agglutination or clumping. So let's say that we have here a positive test, and let's say that the antibody is targeting O157H7 E. coli. In other words, it's an anti-O157 antibody. And if the pathogen is O157H7 E. coli and the antibody binds to it, we'll get this clumping. However, if the pathogen is not O157H7 E. coli, if it is any other microorganism, we'll get a negative result here, and we'll get kind of a uniform color of the media here on the glass slide.